Alright, welcome back. In this video, we're going to go over the Pyruvate Dehydrogenase Complex. So you're probably wondering, what the hell is this? So think back to when you first learned about glycolysis and the citric acid cycle and the electron transport chain. You learned that glycolysis, well, we also learned, because I made videos on it, that glycolysis makes pyruvate. Okay, that's the end goal, to make pyruvate. Then you learned that the citric acid cycle would be next. Okay, but have you ever thought about it? What enters the citric acid cycle? What starts the actual cycle? If you're noticed that actually the citric acid cycle does not begin with pyruvate. It begins with acetyl-CoA. So how are we going from glycolysis, making pyruvate, to the citric acid cycle? That's not possible because pyruvate does not enter the citric acid cycle. This is where the pyruvate dehydrogenase complex comes in. The goal is to convert what we get out of glycolysis, which is pyruvate, into acetyl-CoA, and that will enter the citric acid cycle. So it's like an intermediate complex or intermediate step. It's not widely talked about, but it's something you should really much know because it doesn't make any sense. We can't go from glycolysis right to the citric acid cycle. So you'll notice, as proof, this is a typical Krebs cycle diagram, and you'll notice there's no pyruvate anywhere. It's because, to start it, we have acetyl-CoA. This is the acetyl group, and this is CoA. Now, the pyruvate goes from the cytosol into the mitochondria, and all this pyruvate dehydrogenase complex is happening inside the mitochondria. The Krebs cycle happens inside the mitochondria. You'll notice in this diagram, I don't mean that the acetyl-CoA is leaving the mitochondria, I mean it's being produced there. I just couldn't write it inside the mitochondria. But pyruvate enters the mitochondria and it gets converted into acetyl-CoA. Now, before we begin, there's one important player that is very unique to this pathway. And this is called TPP, also known as the thiamine pyrophosphate. This is a cofactor. Now, all of this is all complete junk. Okay, we don't care about any of it except this thiazolium ring. That's the only important part of it. By the way, there's a very low chance that you can memorize this entire baddie, right? This is a huge molecule, very impossible. Like, unless you have like an eidetic memory, this is a stupid thing to memorize, right? But what you really need to know is this thiazolium ring. That's the only important part. Everything else is garbage, okay? And you'll notice a theme in this video. A lot of garbage, but one little useful part. Step 1. There's a lot of organic chemistry in these steps, in this complex. We're not going to go too much into that because it's not really important. What you really need to know is the end goal, or what we're getting out of this. So, we have this pyruvate molecule, right? We just got out of glycolysis, we're fresh out of glycolysis, we got pyruvate. This thiazolium ring is going to attack the carbonyl carbon of pyruvate. We have this intermediate molecule, and we are basically left with this hydroxyethyl, TPP. So in this intermediate molecule, all of this is taken out. What we're left with is an OH, the carbon, the methyl group, and the thiazolium ring. Out goes CO2, which we'll exhale. And we are left with hydroxyethyl TPP. This is an unstable molecule, but it's stabilized by the nitrogen here on the thiazolium ring. Now, the reason it's called the pyruvate dehydrogenase complex is because there's an enzyme. It's hidden. I didn't draw it on this, but the reason is because it's doing this entire reaction. And it's called pyruvate dehydrogenase, just like the pyruvate dehydrogenase complex. It's there. I just did not draw it because it's involved in this entire step. So it does this entire mess, catalyzes this entire mess. That's step 1. Step 2, well actually first, we have to go over another cofactor. We have a lot of cofactors. This is lipoamide. There's a lot of garbage here. This lysine is garbage. We don't care about it. We're not really, it's part of the molecule, but it's not really involved in the reaction. All of this is also garbage. We don't care about it. It's part of the actual cofactor we care about, the lipoic acid here. But this part's garbage. What we really care about is this ring here with the 2 sulfites. So this disulfide bridge here is what we're really concerned about. So this ring part is what we care about. We don't care about this part, we don't care about this part. Makes it kind of simple, which is nice. Step 2. I know it looks really complicated, but it's actually kind of simple. So here what we're doing is we're basically need to transfer this hydroxyethyl group that we just created and put it onto this lipoamide part. Okay? So this was the original TPP, right? Still attached. What we're going to do is we're going to break it. That's what we're going to do. So this unstable carbon is going to attack the sulfur. Okay? And it's going to basically break this disulfide bridge we have here. Okay? Then we have this intermediate part. This breaks apart. And what we're doing is we're basically separating from TPP. So that diazomium ring, we're separating it. Okay? So we regenerate this. This goes and goes goodbye. We don't care about it. But what we're left with is this part right here. Now the hydroxyethyl, so this is the hydroxyethyl. This gets converted into an acyl group. So this is a double bonded carbon here. With a methyl, this is an acetyl group. And this gets joined to the lipoamide. Okay, I didn't draw the rest of the molecule, right? So notice that this is still there, right? This is it's the entire molecule here. I just didn't draw it for simplicity because the reaction's happening here. Nothing is really happening here, but it is still part of the molecule. We don't forget about it. It's still there, but it's not really participating in the reaction. Only this part's participating, so that's why I only drew it. But there's the rest of it, right? It's still all the garbage. It's still there. So now this acetyl group is joined with this, a lipoamide molecule. Okay? That's it. Step 3. This random CoA molecule, called coenzyme A, will attack the acetyl group. And it will basically break away from the lipoamide. So this is now free, and it's now fully reduced. And now the acetyl group is now joined with the CoA, making acetyl-CoA. That's it for the main part of it. There's actually steps 4 and 5, which will be next video, but that doesn't really involve acetyl-CoA. It actually involves a lot about the lipoamide and FAD+. Not the biggest concern, because we actually got what we needed here, but there is some other stuff that's going on, which will be next video. But since there's a lot of content here, I just want to focus here. But now we got our acetyl-CoA, and this is what's going to enter the Krebs cycle. And that's it. If you found this video helpful, please like and subscribe. Until next time, later.